Hi there, Dr. Dan Guerra from VerEvMed Studios on this beautiful 7th of July 2018. Today we're going to go ahead and get back to one of our original arcs that were going on way back in springtime, which involved aging. Human aging, of course, not just aging of the entire planet like shown here in this very interesting sidelong slide somewhere in Utah, probably in some canyon area. I'm trying to focus in on something about aging that I think needs to really be addressed. That is, we need to examine how aging is very specific to the individual. You can describe it in general terms. You can describe all the endophenotypes and exophenotypes associated with aging, what the environment's like, what the genetics are like, what the epigenetics are like, what the immune system is like, all that, like in diaeventolgy, which is what we're going to focus on today. But I think it's really important that when we look at the scientific literature, which is what we do here at VerEvMed, that you understand that the papers that are being published often are associated with animal models, mammalian models like mice and rats. And those models are only suggestively procured toward human studies. That is, they point in a direction, but they don't necessarily prevail or avail ourselves to an understanding of what happens to the human condition with aging. Aging is different in humans than in other animals, obviously. We live a lot longer than most mammals and not as much as a few others. But that is only just scratching the surface. To understand aging, you really have to go in depth to the individual interactions that occur at the genetic level, epigenetic level, etc., like I've been saying about diaeventology. And that's what I'm going to try to kind of focus on today. So I'm titling this talk Aging Dangerously because aging is dangerous, because living is not something that is an absolute procurement. There are many pitfalls and occurrences in the desert that can cause life or either to end abruptly or to end in some kind of chronological damage, such as in morbid diseases. So how does this happen? And are these diseases related to the aging, and in what way? Of course, there's good arguments in going from one direction to the other, and we'll look at them right now. That's me. After having a lot of coffee, I am definitely a person that promotes coffee use, particularly the kind that's caffeinated. And my name is Dr. Daniel John Guerra. And there's my email address that you can contact me directly about these VerEvMed lectures anytime you choose. And I will very likely get back to you within 24 hours, unless I'm preoccupied with something else, which might mean it could take a bit longer. My company is VerEvMed. There we are at we are at www.verevmed, and there is our email address to directly contact us at the company, info@verevmed.com, right there. That's our beautiful logo. And this is my Facebook address. And my Facebook address, I show you that not because it's Facebook. I'm not promoting that in any way, but because it's a very convenient vehicle from which to launch these VerEvMed lectures. That's where they tend to reside. That's where I place them. They also end up on my Twitter account, which I never really look at. I just can't get into the Twitter thing. And as I've been saying now chronically, that eventually I'm going to get all of these located and referenced and cross-checked according to title and according to content on the VerEvMed website. I just need to have some help, hint, hint, and then I could get that accomplished. All right. So with all of that out of the way, aging dangerously. Do caloric restriction and rigorous exercise correlate with a healthy longevity? A diaeventologic focus. All right. First of all, what do I mean by that? What's the diaeventome? It's the interaction between the genome, the environment, and then the supervening, or we could call it transcendental, immunoepigenome. When I say transcendental, I'm not talking about something spooky and ethereal, just like metaphysics doesn't mean something otherworldly. Metaphysics just means everything but the sciences, including the natural sciences, physics, which was the only real science that was understood in Aristotle's time, hence the metaphysics, which he never coined anyway. Likewise, when I say transcendental, I'm talking in the philosophical sense. Transcendental means above all the other parameters, overarching, right? That condition or the means by which we understand all other things. That's the transcendental method. So that's what I mean. Nothing spooky or mysterious about it at all. So, very briefly, what are we talking about here? I want to say that biological networks are processes becoming eventual in nature as opposed to substantial and fixed. That's why I emphasize event ontologies rather than substance ontologies. Okay? Again, philosophical terms, but I think you get the point. That is an event ontology that reaches across, that's why I have a dia in there from dialectic, the physiological network of genetic environmental interactions. And it's articulated and synthesized through the third component, this epigenetic modification of the classical immune system, thus creating a responsive plastic and elastic memory. Plastic means it's indelible, it's retained after the action or agency. And then, of course, elastic means whatever has occurred can be reversed and can occur again. That's the difference between plastic and elastic in biology. And it's a field capable of biological learning, ideation, imagination, understanding through time. And when I say biological, I mean that the body adapts as well as the cognition adapts, right? So the overall body adapts to these interactions. And that's why it's an event rather than a substance. This is a pattern of events, and it appears as energetic fields. So it's energy, not just matter. So rather than considering particles of matter, think about wave theory of energy. Much like chemical bonds. Chemical bonds aren't substances as much as they are energy fields. And so everything is based on that. The chemistry is obviously embedded in biochemistry, and biochemistry is the foundation of living systems. We can also consider it as a central individual agentically interacting. That is an individual. That is how your biomedical profile would look, as an individual, not as a group, not as a cohort, but as an individual. So central individual agentically, that means upon your own volition or will, through the events obtained across the inherited genome, the changing environment, and again, the transcendental, my fun word there, immunoepigenome through time. So again, you get the idea of event, things constantly changing through time, movement, reaction is what I'm trying to get at here. That's the whole idea of the diaeventolgy. And so I'm trying to use this approach, right, this way of looking at it, so that we can look at the scientific literature and be able to get a larger, more maybe encompassing and engrossing picture, an image of what's going on in biological systems. Again, by using scientific literature. So again, diaeventology, not to press the point too much, but to make it clear to those of you that haven't heard this before. And I'm constantly modifying the definition, so bear with me. It's an adaptational, processive scientific accounting of the physiological, rational human condition through cellular and molecular event ontology. It's the immunoepigenomic induction of neural, endocrine, and metabolic response to the micro and macro environment through classical constitutive surveillance and acquired effector cellular and humoral defense stratagems, such as in the immune system. And it uses reversible, covalent modification, ionic bridges, and hydrophobic interactions of nucleic acids, proteins, and lipids, the biomolecules. It's a general molecular theory for a free will driven, and that's important when you think about individual biomedicine, agency-based, individual adaptation, and knowledge acquiring physiological rational mechanism that better explains, I believe, the core event ontology of human existence, including health and well-being. It's where you put yourself and how you derive your environment through your living system that ends up being your biomedical condition, which is an event, not a substance. That's the point. What are some of die-events in physiology and pathophysiology? Well, they're very straightforward. They're molecular and cellular events that occur across the mechanisms that we look at in biology, and they result in real-time modifications to changing conditions. That would be your environment, both endo, inside the body, and exo, outside of it. What mechanisms are we talking about? Replication, recombination, and repair of DNA, transcription and alternative splicing, and of course translation of RNA, signal transduction, membrane fluid dynamics, cell-fate repertoire such as autophagy, apoptosis, senescence, and necrosis, developmental programs, which of course include embryogenesis, and then aging itself, and indeed at the endpoint, death of the system. Also cellular division and differentiation, including adipogenesis, neurogenesis, myelocytogenesis, lymphocytogenesis, gametogenesis, oncogenesis, and all the other genesises that we might consider in biological systems. That is, things that are generated. Okay. Paper we're going to look at today, one of them, is in the Journal of Biological Chemistry, published in April of last year, 2017, just about a year and 3 months ago. Key proteins and pathways that regulate lifespan. Paper was published by Pan and Finkel, and there's your citation. That is a review article, and it leads with a hypothesis that aging is a risk factor in major human disease, such as cancer, cardiovascular disease, and neurodegenerative pathologies. So remember, it's saying it's a risk factor, the aging itself is. You could also argue, I think, though, that these diseases enhance and promote aging via morbidity and mortality. So I think you need to look at it from both perspectives. You can't just look at it from it being a risk factor. In fact, it's quite possible diseases are the things that are promoting the aging process itself, full stop. You can ask the question, is there a conceptual means to separate pure senescence, cellular aging, written large, sensu stricto, very specifically, from chronic disease? And if so, can aging itself be a target, just aging itself, that process, for biomedical intervention? And that's getting back to what this paper is trying to ask. So I want to combine genomic, environmental, and immunoepigenetic-based paradigms from basic and clinical research. That's the diaeventological focus. And it should be core to the endeavor of analyzing whether or not aging is a risk factor or whether or not it's caused by diseases themselves. All right, from his paper. This is a JBC paper, and we're looking at the epigenetics of aging, right? Now, there are proteins called sirtuins, which I've talked about many times in these various VerEvMed lectures, and I'll go back and I will recap what they are. Sirtuins are NAD, that's nicotinamide adenine dinucleotide, dependent enzymes. They catalyze post-translational modification of histone and non-histone proteins. Post-translational means after the protein's made, of course. Besides the NAD-dependent deacetylation, removing acetate, covalently modified protein, sirtuins can also catalyze demyelination, desuccinylation, and depropionylation. Those are all carboxylic acids and hydrocarbons that are associated with covalent modification of enzymes and other proteins that modify their activity. Sirtuin enzymes are linked to aging. This has been clear in the literature. In fact, here's some example. Overexpression of sirtuin-2 was sufficient to extend replicative lifespan in the yeast S. cerevisiae. Life-extending benefit of caloric restriction also in that yeast, model organism of course, was demonstrated to require SIR2. If you delete SIR2 via homologous recombination, via gene knockout, you don't get any kind of benefit from caloric restriction in yeast. So that means they're linked. Sirtuin-2 activity tanks the formation of extra chromosomal ribosomal DNA circles, which is a unique feature of yeast. Those are associated with yeast aging, in fact. Perhaps the reason SIR2 activity is necessary for that aspect is it could promote asymmetric distribution of damaged proteins between mother and daughter cells. Now, if it controls that asymmetric distribution, it could mean that daughter cells would benefit and mother cells would then go ahead and you see senesce and die, because you have damaged proteins. And these ribosomal DNA circles are associated with generating those fragments, peptide fragments, proteins. In higher organisms, leaving yeast now, other potential mechanisms for life-extending effects of sirtuins include changes in mitochondrial function and biogenesis of the mitochondria, suppression of inflammation, a big key factor here, and the regulation of genomic stability, back to DNA. Right? We have to also consider that in pharmaceutical sciences, there are compounds known as STACS. These are Sirtuin-activating compounds. And they are chemical compounds having an effect on sirtuins. Gee, okay. And of course, sirtuins are a group of enzymes that use NAD to remove acetyl groups from proteins, among other things that we just covered. They are caloric restriction mimetic candidates, these STACS. They are molecules able to prevent aging-related diseases like Alzheimer's, diabetes, and obesity in model organisms. Can't really say that these have been proven by any level in human studies. Okay, but STACS are out there in the pharmaceutical repertoire. Again, go back to understanding sirtuins. They are deacetylases. That's their main understanding and their biological function. And they're activated by various organic molecules. You can imagine a protein being activated by organic molecules. What's the surprise, right? So the NAD-dependent deacetylase SIRT1 is implicated in the prevention of many age-related diseases such as cancer, the neurodegenerative Alzheimer's, and type 2 diabetes, which is a chronic disease of often obesity. Not always. At the cellular level, SIRT1 controls DNA repair and apoptosis, circadian clocks, inflammatory pathways, insulin secretion, and mitochondrial biogenesis. That's quite a lot of biological activity, right? Yeah. Naturally occurring STACS are found, such as the phenolic compound resveratrol. And there are a lot of chemically unrelated synthetic STACS that activate SIRT1 in vitro by lowering its peptide Michaelis-Menten constant, that is lowering its affinity for its substrates. So if it lowers the affinity, or it increases the affinity, it lowers the effective concentration necessary for the enzyme to function, you see. So the functional aspect of the enzyme range is increased when you lower the Km. That's what that means. You could then produce pharmacological changes consistent with SIRT1 activation by using these compounds. However, the legitimacy of STACS as direct SIRT1 activators has been widely debated. That's because STACS can have pleiotropic effects in biology, such as resveratrol itself. Previous studies, STACS increased SIRT1 activity toward fluorophore-tagged substrates. That's how the experiment is done, looking at how the SIRT1 activity responds to fluorophore-tagged substrates so that you can measure it. That's all it is. But guess what? It's not always the case that you get SIRT1 activity increased by STACS to corresponding non-tagged peptides. So there's a problem with experimental design sometimes in these papers. One explanation was that the STACS were binding to the fluorophore-linked substrate, which of course wouldn't occur in vivo because there aren't any. Alternatively, the fluorescent groups themselves might mimic a property of natural substrates. So there's a lot of issues with those kind of experiments, and experiments like that are profound in the animal models using fluorophore because it's an easy way to quantify the measurement of SIRT activity. A big problem with that maybe. Now, let's get into some more biology. Caloric restriction, big key factor in research in aging. Not only does caloric restriction extend the life, but it also seems to prevent or delay the severity of some age-associated diseases. Wow. Okay. Is this going to link back to sirtuins? Yes, it will. Don't worry. Some effects of caloric restriction may result in more harm than good, however, such as malnutrition. So if you have a very defined diet and you restrict calories, you may be also restricting essentials in the diet, such as vitamins and other kinds of substances in the body, just plain protein, for example. If you restrict that, you can be malnourished. And malnourishment is not considered health promoting, nor is it going to increase your lifespan. Now, when that's practiced outside the laboratory, caloric restriction, which is what happens when people do it, you might also enhance the environment for contracting infectious diseases because you may impact negatively the immune system. So it's very important to consider. Mechanism involves significant alterations in energy metabolism when you think about caloric restriction. Response to oxidative damage, whether or not you can carry out that removal of reactive oxygen. Insulin sensitivity, really important in human biology. Inflammation and changes in the neuroendocrine, paracrine, and reproductive systems. Again, a lot of activity there for caloric restriction. The changes are likely triggered by metabolic responses, regulators in the cells, which can sense caloric intake. Your body senses caloric intake. That's because your entire repetitive response, the need to take in nutrition and water for that matter, necessarily is induced when there is caloric restriction. So it's a signal in metabolic pathways to change those metabolism, to conserve, basically. That's what autophagy often does. And that's going to affect, as you might guess, aging itself, aging outright. It could actually enhance it, that is, promote a faster rate of aging during that process. But also it can cause the possible potential contraction of disease. Or it can work the other way. It might be possible that aging is slowed during times of metabolic restriction because of caloric restriction. There's a couple of things very important there, and they are plus and minus. Right? Among the regulators are the sirtuins. We've already encountered them. We already know what they are. I'm not going to tell you again, but there they are, ATP-dependent enzymes. First discovered in yeast, right? There might be a link, though. We're trying to find biomarkers to link caloric restriction with aging or lifespan or disease. Sirtuins may be a good thing to look at. That's why I keep on bringing them up in so many VerEvMed lectures. I think they're fascinating enzymes. Of course, they contain homologous domains that bind NAD. Mammals have 7 of these enzymes. They're called homologs. Sirts 1 through 7. 3 of them, 1, 6, and 7, are found in the nucleus. So they're doing something in the nucleus, probably with histones. Certainly with some aspect of chromatin or RNA metabolism. 3 of them, Sirts 3, 4, and 5, are in the mitochondria, maybe with involved in bioenergetics there. It's mitochondria involved, and amongst other things. And then there's one of them, Sirt 2, that's found in the cytoplasm. And there you might loosely say, well, maybe that's involved in signal transduction or signaling. Sirts 1, 2, 3, 6, and 7 can catalyze deacetylation of protein substrates. Good, because that's what normally they're considered. Sirt 4 has not yet really been shown to be a deacetylase, at least not in biological systems. But it does have, very interestingly, an ADP ribosyl transferase activity. And ADP ribosylation of proteins modifies their activity in unique ways. Sirt 5 was recently demonstrated to have that desuccinylase activity and demyelinase activity. And those are the most robust activities for Sirt 5. And they have very weak, Sirt 5 isoforms have very weak deacetylase activity. So that means removing succinic acid and malonic acid from enzymes or other proteins. Succinate and malonate are important carboxylic acids in intermediary metabolism. Both of them are found in the TCA cycle, for example. That's critical to understand. So here's from an annual review paper of 2013. It's some 5 years old now, but it's still an excellent review article. I suggest anyone that wants to know something about aging and caloric restriction, take a look at that entire series in the annual review. So caloric restriction, okay, and the red-colored arrows are downregulation. The yellow ones are upregulation. And the gray-colored arrows are basically, no one really knows if they're down or upregulated. And the blue coloration, those arrows, are specifically caloric restriction effects. So you restrict calories, you turn up sirtuins, AMP kinase, you alter TOR and insulin signaling. What that does is it shuts down reproduction, and also it may affect resistance to infection. Okay, negatively, that is. It may downregulate it. Okay, it's not clear. Actually, because there are blue arrows pointing to it, it could mean that it has a positive effect. It depends on the system, depends on the diaeventolgy. It does, of course, affect metabolic rate because AMP kinase and mTOR and insulin affect directly metabolism. That's their key feature. And it seems that they promote DNA repair, and they also promote, caloric restriction promotes aggressive behavior in animals. Believe it or not, DNA repair and aggressive behavior, or living as a dangerous organism, actually promote health and longevity of the individual organism. Also, of course, you got to consider cancer and mood and behavior. Those cancer and mood and behavior also are, of course, going to negatively impact health and longevity. Right? So metabolic rate is going to promote DNA repair and aggression. Okay, that's upregulated. Where it's not, it's going to downregulate those agents and those mechanisms that control cancer and mood and behavior, negative mood, negative affect. And those tend to be imposing health and longevity. There's just the slide showing you that proteins are in the mitochondria, the cytoplasm, or the nucleus. Out of all the SIRTs, SIRT1 is the most studied. It influences differentiation of preadipocytes by repressing the peroxisome proliferator-activated receptor gamma. That's PPAR-gamma is a really important transcription factor for controlling lipid homeostasis, which is, of course, important in disease and overall homeostatic control of the human system. Additionally, SIRT1 deacetylates and thus activates that PPAR-gamma co-activator, PGC-1-alpha, which in turn can activate mitochondrial biogenesis as well as induce nitric oxide synthase expression, particularly the endothelial nitric oxide synthase, which further regulates metabolism via guanylate cyclase type of mechanisms. Activation of SIRT1 also causes deacetylation and activation of peroxisome proliferator-activated receptor alpha to turn on genes required for beta oxidation of fatty acids. So this is happening here is SIRT1 is turning on basically an autophagic repertoire, right? It's making more mitochondria. It's inducing the breakdown of fatty acids rather than glucose. Okay. Summary, those changes activate oxidative metabolism in muscle tissue, fat cells, and in the liver. They improve insulin sensitivity because you decrease then the levels of circulating glucose this way. Prevent progression of metabolic age-associated disorders such as diabetes. For example, in the fasting liver, SIRT1 controls pathways that are responsible for the upregulation of gluconeogenesis. Now, wait a minute. Didn't I just say that it enhances insulin sensitivity? Yes, but you see, at low levels of glucose, what happens is fatty acids are oxidized primarily in the major central organ liver. The oxidation of the fatty acids is going to do what? Well, it's going to generate energy, and that energy in the form of ATP is going to be used to make glucose. And the liver will then become gluconeogenic, send glucose back out to the system. In fact, in the early stages of fasting, gluconeogenesis is turned on by the transcription factors CREB and CRTC2. Those further activate, that is, they activate the transcription of SIRT1, which turns the whole thing on. However, SIRT1 activation triggers deacetylation and subsequent ubiquitination and degradation of those transcription factors. So it's a feedback control, ultimately lowering the expression of the CREB targets, including SIRT1 itself. Feedback control, always. At the same time, SIRT1 deacetylates, and we've already talked about this, and therefore activates PGC-1-alpha, which is the effector molecule of PPAR-gamma, remember, and also the FOXO transcription factors to turn on genes necessary for response to long-term fasting. The formation of ketone bodies is what's triggered here. So this is just showing you what I just said. SIRT1 is going to deacetylate, that's CRTC2, which is going to then eventually cause a drop in SIRT1 expression. Right? It's going to shut itself down. But in the meantime, SIRT1 is going to deacetylate PGC-1-alpha and FOXO. It's going to turn on those genes, gluconeogenic enzymes. There you go. Right? And of course, directly, CRTC2 and CREB2 turn on gluconeogenic enzymes, but this is going to be shut down because you're going to deacetylate that and turn it off. Right? Lovely feedback control at the enzymatic metabolic pathway level. Sirtuins also play an important role in controlling inflammation, which contributes to numerous age-associated diseases. More inflammation seems like an enhancement of aging, and of course, diseases themselves. Again, how do you separate the two? In fact, both SIRT1 and SIRT6 suppress the activity of a key activator of inflammation, which is called an NF-kappa B, nuclear factor kappa B. SIRT1 can directly deacetylate one of the subunits of NF-kappa B, called P65, and that reduces the ability of NF-kappa B to activate the transcription of pro-inflammatory genes. So basically, it shuts down inflammatory gene synthesis. Pro-inflammatory cytokines, for example, and growth factors. SIRT6 deacetylates histones as well at the promoter region of NF-kappa B of all those regulated genes, and that represses them and thus suppresses inflammation even further. So it works on histones as well. So it works on histones, and it works on that P65 unit, subunit of NF-kappa B directly. So two different levels. SIRT1 and SIRT6 may work in concert then, as SIRT1 can activate SIRT6 transcription by deacetylating and therefore activating the transcription system FOXO3A and RF1 complex on the SIRT6 promoter. It's lovely the way these here. So here you've got it where SIRT1 works on the deacetylation of this FOX3O, which then turns on SIRT6 expression. SIRT6 expression then deacetylates those histones and deacetylates NF-kappa B, which then shuts down the synthesis of the pro-inflammatory cytokines, working in concert. Mitochondrial sirtuins, on the other hand, govern key aspects of energy metabolism, especially during prolonged fasting or caloric restriction. In addition to its role in fat oxidation, SIRT3 promotes the catabolism of acetate and amino acids. For example, it deacetylates glutamate dehydrogenase, which converts glutamine to alpha-ketoglutarate, of course. And alpha-ketoglutarate then can be used in the Krebs cycle for energy production. Okay. Another recently identified target of SIRT3 is lysine 88 of ornithine transcarbamylase. These are again enzymes you find in the mitochondria. Deacetylation of that enzyme by SIRT3 activates the urea cycle to dispose of ammonia. So amino acid metabolism, particularly amino acid catabolism, can promote carbon movement into gluconeogenesis. So that's essential when energy is produced by amino acid digestion, right, or catabolism. Another mitochondrial sirtuin, that is SIRT5, can also activate the urea cycle by deacetylating or more likely desuccinilating, because it's SIRT5. Remember, that's its most common biological activity, enzymatic activity. And it desuccinilates carbamyl phosphate synthase 1, thus promoting urea cycle. That is removal of toxic ammonia because you're already catabolizing amino acids because that's what you're doing during fasting. Yeah. SIRT3 also deacetylates HMGCS2, that's 3-hydroxy-3-methylglutyl-CoA synthase 2, and that's, of course, essential for ketogenesis. Okay, so you're doing ketogenesis, you're doing amino acid degradation, and you're doing gluconeogenesis, and you're doing urea cycle to remove ammonia. That's what happens during the fasting state. Acetyl-CoA synthase 2, which is necessary to move fatty acids into the mitochondria for beta oxidation, is also tightly controlled by acetylation, and SIRT3-mediated deacetylation on a particular lysine C635 of the acetyl-CoA synthase, stimulates this activity. So this is all working beautifully in concert. One target for SIRT3 control of beta oxidation is also lysine 42 on the long-chain acyl-CoA dehydrogenase, a particular frank enzyme, meaning that is its specific enzyme in beta-ox. Interestingly, SIRT3 activates mitochondrial pathways that mitigate oxidative damage, that is, that turn down. For example, SIRT3 stimulates the activity of an enzyme called isocitrate dehydrogenase 2, and that drives the glutathione detoxification system and the activity of superoxide dismutase by deacetylating lysines 53, 68, and 122 of that isocitrate dehydrogenase 2. Basically, you generate NADPH, and NADPH drives a glutathione detox system. That's what ICD, isoform 2, does. It makes NADPH. Because SIRT3 also deacetylates components of electron transport chain, it may actually reduce or slow, make more sluggish the potentiation of reactive oxygen because of hyperactivity, for example, of the ETC, electron transport chain. This is basically summarizing all of that. Okay, so it's going to deacetylate. This is the synthase. This is the beta oxidation enzyme. This is the urea cycle, the ornithine transcarbamylase. There's the IDH2, helping control ROS or protecting against ROS. Remember, you're going to get ketone production, amino acid metabolism, and more ROS production because of SOD2 deacetylation. So SIRT3 is really a good player in the mitochondria. So hopefully I've convinced you that Sirtuin-activating is really important. And if you've got a biological agent like resveratrol, that phenolic compound you find in grapes and in berries, certain berries, could be correlated with extension of lifespan. In fact, they've been shown in yeast and in worms like C. elegans and in flies like Drosophila. But there's no consensus in mammalian systems. It doesn't always seem to work that way. There you go. Model organisms only go so far. Resveratrol, for example, doesn't increase longevity in mammals, but it is associated with altering the morbidity of metabolic deficits associated with normal aging or diet-induced obesity. So it does seem to have a positive effect, but it doesn't seem to have a direct effect on aging. So you're starting to tear the two things apart, separate them. So remember that STACS controls sirtuins. They kicks up sirtuins. They increases mitochondrial function. It increases genomic stability by enhancing DNA repair, decreases inflammation, changes metabolism, beta oxidation, amino acid oxidation, increases stress resistance. Rapamycin, which is another compound that's often used, which of course binds to the target of rapamycin, which is a kinase which is anabolic. Okay, this is opposite of what this repertoire of sirtuin is going to do. But when rapamycin binds to mTOR, it's going to tank inflammation. It's going to decrease protein translation. Going to increase autophagy. Going to change metabolism back to this beta oxidation pathway. It can also enhance stem cell function by altering stem cells to become morphologically associated with changes in cell structure to mimic the cells that are necessary for use, such as those cells for autophagy, for autophagic systems. And it's also going to tank ribosomal biogenesis because you're not in anabolic mode. Fasting also, so here's fasting. See these three components: rapamycin, antibiotic, tanking mTOR, STACS, enhancing sirtuins, and fasting itself is going to drop what? The insulin growth factor 2. Okay, so insulin growth factor signaling is really important here. And when you tank insulin growth factor stimulation, you increase stress resistance, you change metabolism, beta oxidation, and it's also been linked to a decrease in cancer. This is from a paper published in JBC in 2017. So, same paper. In general, the inhibition of mTOR, there's the complex with the various also associated proteins like Raptor, Deptor, PRAS40, and MLST8. Those are all proteins that are associated with the mTOR complex. That's the mTORC. The C is for complex, you see. All right, so what happens then is that amino acids and growth factors can all regulate mTOR. And when it's on, it's not what you want, right? So you want to inhibit it. Because when you inhibit it, then you decrease ribosomal genesis, you decrease lipogenesis, you decrease pyrimidine synthesis, which would make more DNA. Tanking it would then, of course, tank the entire system here so you don't get translation, right? It would enhance autophagy because tanking mTOR would remove the suppression of TFB and ALK1. Therefore, autophagy would increase. Likewise, here, tanking it would decrease the amount of the pro-inflammatory interleukin alpha, which would decrease the amount of NF-kappa B. NF-kappa B is associated with senescence-associated secretory proteins. That response is also the senescent aging response is considered negative because it promotes aging in very complex ways, which I discussed in at least 3 previous VerEvMed YouTube videos. The SASP system. All right. I get from a JBC paper, this diagram. There's a syllogistic acetylation pattern in aging, and these are metabolic dia events. Although mTOR activity declines, sirtuin activity increases when tissues are depleted of nutrients. Exceptions include various pathways involving AMP kinase that indirectly increases NAD levels, which of course activates the SIRT1. That activity then, sirtuins then deacetylate the FOXO, okay? And then SIRT1 also deacetylates the LKB1 kinase, which then activates AMP kinase, which then triggers the whole response for more SIRT1 activity, okay? So again, AKT, if it's blunted or shut down, here's the insulin growth factor, turning on the P13 kinase, that's phosphatidylmastol 3 kinase, which normally activates AKT. When that happens, okay, that would normally block these systems, which would then activate mTOR, okay? But that's not occurring because the AMP kinase is blocking that. Okay. At the same time, it's turning on the SIRT1, which is super activating the AMP kinase, which is activating FOXO, which again is regulating the entire system. And FOXO is not going to allow for more mTOR activity because it's going to be blunted out by the deacetylation pattern. All right, so all of this functions to tank mTOR, activate AMP kinase, activate the SIRT1 activity. So it's a syllogistic, right? It's a syllogistic. What I mean is this will occur. Oh, okay, A goes to B, and because A goes to B, B goes to C. That kind of syllogism. Okay. Here's a paper from Neuroscience. Neuroscience 2015 paper. Immune-associated aging is mitigated by sirtuin 3 deacetylation of the FOXO3A pathway. Now here we're talking about resident macrophages known as microglia in the central nervous system. So microglial SIRT3 knockdown resulted in an increase in cellular and mitochondrial reactive oxygen and a decrease in the expression of the enzyme that would remove the reactive oxygen, that's manganese requiring superoxide dismutase. Okay. So if you knock down SIRT3 in microglia, you activate a system which increases reactive oxygen, and that's going to cause neurodegeneration. So you want SIRT3 active in microglia. That's what this is saying. The overexpression of microglial SIRT3 boosts the expression of catalase, which breaks down hydrogen peroxide, and increases also the activity the activity and even the expression at the transcriptional level of the manganese-associated requiring superoxide dismutase, which is all good. So overexpression of that enzyme even enhances the promotive effect of a healthy central nervous system. SIRT3 overexpression further induced the transcription and nuclear translocation of the transcription factor, FOXO3A, which in turn, once it gets translocated to the nucleus, transcriptionally activates the expression of, you see, here's the transcriptional control of the manganese requiring SOD and catalase, thus making ROS and potentially increasing longevity, okay, because you decrease the amount of, you tank reactive oxygen by making those enzymes, increasing longevity in the mammalian brain. And that's what this neuroscience paper suggested. So in total, what this is saying is that there is a neuroimmunoepigenetic, it's epigenetic because it's sirtuin, right? Mediated control over senescence. It's neuro because it's in the microglia. Microglia are in the central nervous system, and it's immuno because microglia are immunological cells. It's epigenetic because of the effect of sirtuin, which is altering the acetylation pattern, thus controlling FOX3A, thus activating manganese requiring SOD and catalase, removing reactive oxygen, potentially increasing longevity in the mammalian brain. Here's a paper published in 2016 in Antioxidants Redox Signaling. There is your citation. This paper talks about the manganese superoxide dismutase acetylation and dysregulation due to the loss of SIRT3 activity. So if you lose SIRT3, you're going to have an acetylated enzyme, right? That's going to promote the luminal B-like breast carcinogenic permissive phenotype, which is the most lethal of those kinds of breast cancers, the luminal B-like breast carcinogenic permissive phenotypes, the worst. SIRT3 protects against the most virulent form, then that's the most virulent form of the estrogen-positive breast cancer. That's what luminal B is. So it protects against it. Immunoepigenetic diaeventological mediation of a common cancer leading to death. So see, before we were talking about aging per se, sensu stricto, now we're talking about diseases which would promote the aging, for example, unto death, right? Because you're now ameliorating, you're blockading via sirtuin activity, breast cancer, the most virulent form, which will then involve the endocrine system because these are estrogen positive rather than estrogen negative breast cancers, some of the most virulent forms, this luminal B type. So, wait a minute. We've been saying positive things about it, about how sirtuins work. We talked about caloric restriction. You often get a nod towards saying that Not only is it good to restrict calories, as long as you don't become malnutritive, by the way, of course, but also maybe exercise is good, right? But wait a minute. Aging comes with exercise just in general, right? And there is a lifestyle associated osteoarthritis, which is not good. In fact, that is going to enhance aging. It comes with it. In fact, the more active, the more you're likely to get osteoarthritis. Isn't that terrible? Isn't that a pain? Those of you that know what I'm saying know what I'm saying. There is a link between pro-inflammatory mediators and epigenetic modifications of the osteoarthritic joint. There it is, the damaged osteoarthritic joint, and there is the normal, healthy one. These are normal chondrocytes. These are abnormal chondrocytes, right? Or also known as hypertrophic chondrocytes. They're getting larger, and they are responsive to and cause an inflammatory mediation, which causes the osteoarthritic joint. So there's a cycle of inflammation generated due to a recurrent release of cartilage-degrading proteins like metalloproteinases, matrix metalloproteinases, and of course pro-inflammatory cytokines. And there's an aberrant epigenetic modification like what? Take a look. Look at all these cytokines, TNF-alpha, interleukin-1 beta, IL-6, IL-8. They all are epigenetically controlled. How? DNA methylation, histone modification, usually that's acetylation, and microRNA expression. All of those are epigenetic control over cytokines. Look at here is iNOS, the inducible nitric oxide synthase associated with osteoarthritis. What do you have here? You have DNA methylation controlling it as well as histone modification. Proteinases, like I told you, the metallic matrix proteinases. There's 3 and 9. There's ADAM, which is, of course, associated with Alzheimer's disease. A beta protein degradation and amyloid plaque. That's the ADAM system I talked about before. There's cytoplasmic proteases, extracytoplasmic matrix proteins. Those enzymes there controlled epigenetically. Chondrocyte growth factor gene is controlled epigenetically via DNA methylation. And there's all these transcription factors which are going to run this course of disease as well. This came from a paper in Frontiers of Bioenergetics, Bioengineering and Biotechnology, published just about 3 months ago in March of this year. There you go. So a lot of epigenetic control, you see? So there's a pathological progression of osteoarthritis, which comes with exercise, which is linked to just aging. It's particularly lifestyle associated. The more exercise, the more potentiation of osteoarthritis, all epigenetically controlled, pro-inflammation, thus causing this hypertrophic response, thus inducing this osteoarthritic joint. Okay. So you get what's called a hypertrophic state, further causing damage. It's a nasty cycle, enhancing all these epigenetic phenomena, enhancing inflammation. So is exercise always positive? Well, no, because aging, just normal aging, means more exercise, right? You're using your joints more because you're moving around. You're getting up and maybe you're going to make dinner, or maybe you're taking that walk, or maybe you're playing tennis, or maybe you're cutting down a tree in the woods. All of that is going to enhance osteoarthritis. And so even though you're very biologically active and physically active, you can get more and more osteoarthritis, and all this is epigenetically controlled. And wow, isn't that kind of a downer when you think that, hey, the more active person is going to live longer? They may live longer for other reasons. Who knows? We'll get to that in a minute. But you also can get a lot of joint damage and pain. Now, studies of dietary restriction and the genetics of aging have revealed a close connection between nutrition and aging. Okay, I want you to know that the papers, the abundancy of them in the literature. Capacity to rapidly adjust to inevitable fluctuations of the food supply, of course, is what dietary restriction is sensing. Those fluctuations in food supply, because we're heterotrophic organisms and we're omnivores at that, and we have essentials at that too, such fluctuations can lead to the evolution of a marked phenotypic plasticity, of course, that allows an organism to focus on physiological states geared for growth and reproduction when there's ample food, and to postpone reproduction. That's a system called diapause, most commonly studied in insects, actual. Diapausal ectasis, as well as what's shown as pupation, for example, in insects, something I studied as a youth in my previous graduate studies. And that happens when food is scarce. Major mediator of this plasticity is growth hormone IGF-1 TOR pathway, okay, insulin growth factor 1 TOR pathway. And that all links up with lipid metabolism. Mutations in that system, the growth hormone IGF-1 TOR, that increase rates of biosynthesis in early life, okay, may enhance fitness by accelerating growth in early life, like that is prepubescent or right at puberty, or maybe early adulthood. That increases reproductive output, which is when reproduction should occur, because that's when the haplogenome is more nascent and therefore less prone to mutation, right? Early rather than late reproduction. However, mutations which affect that pathway positively, activated that is, can also increase something called hyperfunctionality, which can exacerbate pathologies caused by hypertrophy and hyperplasia, which are going to happen because of enhanced activity of growth hormone, for example, or IGF signaling or mTOR activity. And therefore, that could all accelerate hypertrophy and hyperplasia, can accelerate aging. Now, hyperplasia, whoa, look out for that, put an asterisk next to that. That could also be associated to, a lot of cell division, and aberrant cell division can, of course, lead to tumorigenesis. Look out for that. By that view, poor maintenance does not cause the demise of the soma, the body, rather excess biosynthesis does. So it's not poor maintenance that's having an effect here. It's because you have excess biosynthetic pathways. This is hypernutrition. That is, the soma is not disposable, but bloated, okay, because you have a lot of body mass. Thus, the hyperfunction theory of aging can effectively link a trade-off theory, that is trade-off in age, with empirical findings about the control of aging rate by growth hormone, IGF-1 signaling and mTOR, thereby generating a new integrated account of the causes of aging. So this whole thing about hypertrophy and hyperplasia because of hyperactivity, right, is not necessarily a good thing, right? So here's a general review of exercise science and longevity. I just put it out here very, very succinctly, because otherwise you'd go into the muscularity of it, which would be hundreds and hundreds and hundreds and hundreds of papers, which we're almost at the end of this talk, thank God. And we're not going to do that. We're going to summarize. Over 400 publications I found just last year concerning serum biomarkers, central nervous system neurotransmitters, neuropeptides and growth factors, and all their receptors, active humoral and cellular immunity, almost said humanity there, and decrease in the loss of lean body mass with concomitant low body fat indicate from model animal studies, mice, for example, and rats, where active regular exercise is imposed, okay? All those studies seem to indicate the lifespan and cognitive capacity. Yes, they study cognitive capacity in rats, like in the maze and whatnot, and burying marbles and strange things like that, neuropsychiatric legerdemain, are increased with regular exercise. Okay. Okay. Now, animal models, okay, but what about research on human subjects? Far less reported, believe it or not, except for lots of short-term interventional studies. Like, let's put people on a diet and make them more active by getting on a treadmill for 6 weeks or something. But in general, favorable outcomes are also suggested in those in those human studies. It's still suggested, but the kind of research that's done there is not nearly as controlled as in the animal studies. But it does say regular active exercise and good nutrition, including caloric restriction, seen to promote healthy physical and psychological aging in human populations. That's still the paradigm out there. Although, as I say, put a big caveat in that paradigm, careful, controlled studies are actually not all that common. Double blind, for example. In fact, I put an asterisk there, a very recent paper that I heard about on a podcast just this morning while taking my 8-mile walk. Oh yeah, I'm not involved in overactivity. That paper was discussed. Okay, it was the April American Journal of Psychiatry. There's your citation. That paper, which is a meta-analysis of a lot of other papers because it's a review article, links physical activity to suppression of major depressive disorder emergence in a population that's coming from young to old. Okay, but you can ask, is that a consequence or a coincidence? So if people are more physically active, they're more physically active because they have better agency. They're more interested maybe in their lives. they're more interested in their lives, wouldn't that mean that they're not going to be succumbing to depression? Yeah. So it could be coincidental. Coincidental doesn't mean there isn't an association, though. I'm not saying that. I'm just saying it's coincidental. Doesn't necessarily mean it's a consequence, though. Doesn't mean that because you're more physically active, therefore, okay, because of that, because you take long walks up on mountainsides in the morning every day, you're not going to be subjected to or not progress into depression. May just mean that you're not the kind of person that becomes depressed. And by the way, you are a person that's very active. See, so it's coincidence. You gotta taking those things apart is not so easy, even when looking at studies published in the literature. So we have to think about the diaeventomics there. It is clear that individual genetics and the environment that that individual is embedded in, as in Dasein, interacts with the immunoepigenome, and it can play a dominant role in human longevity. There's no problem with saying that. And of course, the environment component is nutrition and exercise, right? That's part of the environment, right? How you live your life in terms of the kinds of things you eat, whether you restrict calories, overall nutrition, and whether or not you work out a lot and exercise through life. So here are a couple of interesting models. This is a central conceptual framework for the biology of aging and encompasses proximate and ultimate causes. Proximate causes like high food intake has a direct effect on aging. You get nutrient sensing pathways which control aging, okay? And that's through, again, this insulin growth factor 2 mTOR pathway. These are intact molecular machineries in the youthful system. That ultimately can lead to damaged molecular machinery or senescence and disease and death because of hyperactivity, okay? You get a lot of off products of metabolism, such as reactive oxygen. You have then a proximal determinant of aging, and that has to do with the production of reactive oxygen. So you get stochastic, that is random damage, which can feed into overall damaged molecular machinery, which can increase disease and alter longevity, that is, hasten death. Okay, what this does do with high food intake, the proximal cause in dietary effects on aging, considering that parameter, is it does shut down somatic maintenance. That is, you decrease the amount of autophagy. And that can also link to antioxidant defense. Okay? You get less of it. The ultimate cause, where this could be the efficient cause, Aristotelian term there, and this is the ultimate cause. This could be something like the formal cause, Aristotelian causes. You get an increased rate of intrinsic mortality. That's overall the ultimate formal cause of what's happening to the system. And evolutionary forces are going to then cause natural selection, and natural selection is going to then try to enhance somatic maintenance and give you more autophagy. So formally, overall, the genetics is going to work against this dietary impact of you choosing to have a high food intake in your early days. Right? So that's the standard model. Aging is a wear and tear process. It's caused by the accumulation of molecular damage to biomolecules like proteins and nucleic acids. Pathways of natural selection affecting aging rate after alter the rate of accumulation of damage. So this is your overarching genetics and immunoepigenetics trying to limit the amount of this damage by increasing somatic maintenance. Now, another way of looking at the alternative model is down here. Aging is caused by a hyperfunctionality or overactivity in late life of biochemical processes. For example, growth spurts. Such hyperfunction leads to hypertrophy and hyperplasia-driven pathologies like cancer, for example, or hypoplagic heart, which is cardiovascular disease often. And natural selection affecting aging rate, again, alters the hyperfunctional level, but back here at the level of insulin-like growth factor and mTOR directly. So rather than working later just on maintenance, not functioning in the proximal causes of standard model, but the alternative model, high food intake, right, has a direct effect on aging. You get nutrient sensing pathways controlling aging. You get lots of growth and reproduction occurring, right, because of high food intake throughout life, right? Hyperactivity throughout, even late life. That's going to give you this hyperfunctionality. That's going to cause hypertrophy and hyperplasia. That's going to lead to death because of cancer and cardiovascular disease, right? This insulin-like growth factor mTOR is going to block autophagy, which would have blocked hyperfunctionality. What natural ultimate formal cause is going to do is block that enzymatic machinery That's the AMP kinase and sirtuin kicking in, right? Yeah. This is like manganese superoxide dismutase and catalase down here. Here is the sirtuins blocking this whole system upstream, therefore removing the negative effects of autophagy. Autophagy then blocking hyperfunction, increasing longevity. So that's the alternative model. Okay. Perhaps if more resources are available, then organisms should invest more, not less, in somatic maintenance, in which case the dietary restriction ought to shorten lifespan. Is that what would happen? But it doesn't seem to occur, right? So there's a problem with the standard model. The hyperfunction theory then looks maybe more promising. It provides a more straightforward explanation. Low food levels are associated with reduced levels of nutrient sensing signaling like mTOR, so you don't get that hyperfunctionality throughout life. That is vis-a-vis later in life. The reduced signaling lowers biosynthetic levels, reduces overall biomass, such as fat deposition, and suppresses hypertrophy and hyperplasia, which is going to, of course, decrease the amount of potential for oncogenesis and cardiovascular disease. The end result then could be an increase in longevity there. Yeah. So, take a look at this. Here's the evolutionary explanation for the mechanism of dietary restriction in terms of standard model and alternative model. Okay. A is the disposable soma theory derived from damage maintenance paradigm. Ample food, lots of food, you get lots of growth and reproduction. You don't control maintenance. You don't keep the body maintained. DNA repair, for example. There's a disposable soma then. The body produces enough of extra tissue that you can enhance longevity, but not very much because you're spending all of your energy in growth and reproduction. Dietary restriction in the evolutionary model of normal disposable soma theory, you get more somatic maintenance, less growth and reproduction. Maybe you're not as reproductive. Maybe you don't grow as much, put on a lot of adipose early on. Enhances longevity. See the darker, deeper arrow. Living longer, not living as long because of disposable soma. The alternative theory, the bloated soma theory, is ample food. You do a lot of insulin, IGF-1, mTOR signaling, hypertrophy. You get the bloated soma, a lot of adipose tissue, for example, obesity, yeah. More pathology, more death, cardiovascular disease, cancer. Dietary restriction, you get less signaling through insulin, IGF-1, mTOR. You get less hypertrophy or none, right? Limited. Therefore, you limit pathology and death in that way. Okay. This comes from the Interview of Physiology. Again, it's published 2015. Great papers. So what are some conclusions we can come from and finish this hopefully stimulating VerEvMed lecture on the 7th of July, 2018? The hyperfunction theory, which is a purposeless continued activity of developmental programs in later life, leading most lethally to diseases of hypertrophy and hyperplasia. Hypertrophy would be cancer, hyperplasia would be cardiovascular disease. That hyperfunction theory provides a clear and simple explanation for many observations within biogerontology, that is the biology of aging. The insulin growth factor 2, along with growth hormone in mammals, plays an evolutionary conserved role in controlling mammalian aging, that is by limiting it via sirtuin activity, for example. And also ghrelin plays a role in growth hormone. I didn't get into that today, but I will in subsequent lectures, and have in previous ones. The adipokine ghrelin controls growth hormone, by the way. Was noted that insulin growth factor 2 affects both growth and stress resistance, but quickly assumed that only the latter was relevant. But maybe growth is more important. In fact, one of the first genes identified as controlling longevity encodes an insulin receptor substrate homolog. The diaeventolgy of gene expression, epigenetic modification, and immune surveillance, like, for example, the microglia analysis we went through, mediate specificity of molecular events, sculpt them, that help direct the eventuality of aging unto death. Again, event ontology. Therefore, the pathways controlling aging, including growth hormone and insulin-like growth factor 1, also control growth, and maybe it's growth itself that causes or that is most significant in aging. Maybe it's growth in inappropriate stages. So it's something to ponder. There's our old friend Albrecht Dürer's Melancholia, the philosopher thinking about all the things she knows. She has all the tools of wisdom. She has the compass for mathematics. but she realizes that the infant doesn't pay any mind to the scales, what's good and what's bad, right? But the thinking melancholia, right? That spirit does think about it. And while she sits there contemplating all this science, right? All the science that's around her, she realizes that maybe she can have no effect at all and biology will win the day. So we're going to end now with what we started with, right? The beautiful Utah expanse, now set straight, not looking so dangerous because we've now divulged an examination of the diaeventological parameters of aging. Maybe we know a little bit more than we did before. There's my email address for the company, info@verevmed.com. There's the website. Please come to it, attend to it, contact us through there, and you will become our clients, and we will work with you in analyzing scientific literature. Remember that we are scientists verifying published evidence in medical biosciences. My name is Dr. Daniel John Guerra. I have a PhD, biochemistry, lipid biochemistry. There's my specific email address. You can contact me, and I will get back with you. So with that, we are going to end our discussion today. And I will end with what I always say. Bye for now.